Thank you for your interest in dementia research. And really, this is a time we've set aside really to explore possibilities for dementia research. As we were gearing up to give this webinar just in the last few minutes, we were talking a lot about population genetics and imaging and how really this is like a new opportunity to bring a lot of these things to bear, really with a whole bunch of international collaborators and really sort of make headway in dementia and other types of neuroscience research. So that's really cool. And I'm really excited to introduce you to the people that are on the panel today. With that, I'd like to introduce the panel. So I'm really excited to have Laura Winchester here. I got to work with her in an event we'll tell you about in a little bit. A couple of months ago, she's at Oxford and really thinks a lot about large-scale genomics. And then my friend Ahmad is here from King's College, and he really has been involved in a number of types of dementia research. He's a clinician scientist over at King's College. And then David Llewellyn from Exeter is here. He's very involved in organizing something called the Dementia Network. He'll tell you about that in a little bit. And then my colleague Ondrej, who I mentioned before, is here from DNAnexus, really an expert in imaging. And Ondrej and I have really been doing a lot of work lately exploring links between genomics and imaging and doing some co-analysis, particularly on the UK Biobank research analysis platform, which we'll talk about just a little bit at the end. So awesome. All right. So with that, maybe we'll jump in, and this is going to be sort of a very informal Q&A format. And we're really sort of trying out this public roundtable format. So if you like it, let us know. If you don't like it, let us know. If there's things we can change, that'd be just fine. And you can always email me or support at DNAnexus if you have further comments, or pop things in community.dnanexus.com if you're a member of that community. Awesome. All right. So I'd like to go around to the panel, and maybe why don't we actually start with David? And he explained this to me once, but we were in the middle of doing a bunch of things. So David, how did you end up working on dementia research, and what's kept you interested? I mean, why are you still interested a number of years later?
Thanks, Ben. I don't know, to be honest. I don't know how I ended up in university. I don't know how I ended up doing a PhD. I don't know how I ended up in Cambridge doing dementia research, and I don't know why I've stuck at it. So I'm not really very insightful. I mean, if I can unpack that slightly, I think it was the worst area of medicine to move into when I did. It was completely drastically underfunded. It was a taboo area that people didn't like to talk about. And it wasn't like we've seen so much change. And it was the poorest of relations to conditions like cancer and cardiovascular disease. And you definitely wanted to do that if you were ambitious. So I think it says more about me and maybe my stubbornness, but actually, really, it was intellectual curiosity. That's all it was. I just thought it was the most fascinating. The human brain was the most fascinating thing in the known universe, the most complicated thing in the known universe. And to understand how things can go wrong and to start to think about how we might be able to improve that, I just thought that was valuable, personally and intellectually. And I've never changed that view. The funding situation has improved, but not by that much. We're still very much the poor relation. There've probably been, this is not an exaggeration, there've probably been more papers published in the last couple of years on COVID than in over 100 years of dementia research. And I just think that's unacceptable and shocking. So that's where I'm coming from. I like working in a field where I think we can make a difference. And there's so much to learn. So if you're intellectually curious, I think you're in the right place.
Wow. I mean, that on a scale of like how interesting I thought that response was going to be, that way overshot. So thank you,
Does
David.
that say something about me? Your opinion of me, Ben?
That's not where I was going with it. But anyway, but I think one thing that must be launching to the front of people's minds is as well. I mean, in addition to being extremely fascinating. I mean, this is a class of sort of a large group, really, of diseases that'll affect something like 50% of humans during their lifetime. And so maybe I could ask Ahmad after he finishes his sip of water to talk a little bit about like the human impact, because he's a clinician and really thinks about that a lot. How did you end up really in dementia research and also working on it in the clinic and doing clinical trials?
Hi everyone. Same like David. I never thought like when I entered medical school, they're gonna end up doing PhD in genetics. Then later on, it takes me later on just doing more of statistics and data science, and most of the time. I always knew that I want to do research. I think the way I thought about medicine is slightly different. I thought about it how clinicians were also studying the disease they encounter in the clinic. But when I went to med school, I didn't see this. I was really disappointed. I wanted to do more, and we were just stuck in protocols, and that was a motivation to go to genetics. I think it's a great time to be a geneticist, and it's a great time also to be involved in data science. Now we have these massive data sets, and we could answer multiple questions in different ways. It's a really exciting time, actually. Probably physics was dominating the 20th century. I think 21st century is now more of like big data and biology, genetics taking over. So it's great.
Ahmad, can I ask a little follow-up question? Like, I mean, do you think that genetics changing the clinical protocols is accelerating at this point?
I think so. It's fascinating. I think with genetic data now we have the ability of for the first time to predict, and that's really crucial in diseases. And with prediction, we could do prevention and also maybe also more of as a personalized medicine. So genetic now is unlocked of this massive data, and now we could study disease in a different way.
Awesome.
A good example for it is the genetic gene therapy, which wasn't possible before. So the idea is like you could target specific people according to their genotype, or like how your genome is fantastic. And now we're taking it also to next stage. We also we do clinical trials, not just focus on a specific group, and also we could tailor their medication according to their genome. So it's fantastic time also to be able to do this.
Yeah, I mean, I certainly agree and hope so. And I imagine most of the people in the audience probably feel the same way. But I'd like to sort of move over to Laura Winchester, who really does a lot of sort of large scale population genetics. And, I mean, thinking about sort of what's your take on how the work you do affects clinical stuff, and why did you end up doing dementia research?
So I've been doing with the genetics data as like Ahmed was saying, but my interest is more in looking at target identification. So using that same genetic data, but rather than prediction, I look for targets, because we're looking for drug targets. Because the dementia field, what we're looking for really is treatments, hopefully. And so if we can use the genetic or the omics data to find new targets, I think that would be key in the dementia research at the moment. So coming in from a different angle, but using same data.
So I'm going to confess that I lied to you, Laura, and I said this is going to be like a really sort of easy, relaxed webinar. And now I'm going to ask you some mean questions about etiology. So that said, I mean, if you're looking for drug targets, you need to tease out etiologies of those diseases, presumably. And so how do you go about teasing out etiologies of these really sort of complex and widespread dementias?
You have asked me a tricky question. Well, that's tricky. So I'm not so focused on that because I don't come in from a clinical standpoint. So I'm dealing with data sets actually similar to UK Biobank. Within UK Biobank, I've been using more all-cause dementia where you can't get so much of that yet. So that is maybe something that I might think about next, but at the moment, that's not quite my specialty. I might defer to either some of the other panelists to answer that one, if that's all right. I'll put myself on the side for a second.
So actually, we have a question from the audience that I think is really follows along well from this, and that is that's about the phenotyping side of things. Does the panel think that cognitive and behavior is well understood well enough for preventing and treating dementia? And are our interventions coming in too late?
We don't know. Like, is the honest answer. I think the failure to intervene at a late stage in clinical trials and achieve disease modification has meant that we hypothesize that by going earlier, we'll be able to make a difference. But there's a real problem that the earlier you go, the more stable people are clinically. So you can go from a moderate dementia where you try and stabilize a patient with a well-established, with very severe symptoms, actually, debilitating symptoms at that stage. Or you can go to early dementia when it's quite mild, or then you can go to the mild cognitive impairment stage where they haven't developed the functional consequences that define dementia. And then you start saying, well, no, we need to go even earlier. But if you go even earlier, then the length of follow-up required in trials to see a clinically relevant outcome becomes longer and longer. So we should say, well, we'll randomize people at birth, and that would be very effective potentially, but it'd be very impractical and very expensive. So we're struggling to define what the sweet spot is, and we're struggling to define what the intermediate endpoint should be in the trials that are more sensitive. The kind of endophenotype idea. But the validation of those in a way which would satisfy regulatory requirements for drug development is a non-trivial question. So what is the sweet spot? Probably people who have pathology, who are in the MCI or even just before MCI stage, who are at the high risk of developing dementia eventually. They won't develop dementia during a trial, and you'll use intermediate outpoints to try and demonstrate a benefit of the drug. But really good question, and it is very difficult, and there are a lot of unknowns in that. So we're way off knowing that with certainty, I think.
I agree, David. I think there's other things to consider, like the cost of this, and the other thing is, which is it's a crucial aspect. The second thing is the ethics. Not necessarily everyone with this risk gene will develop dementia. So how are we going to intervene clinically with possible treatment? There is ethical issue here that we need to consider, and this need to be discussed at a certain point. We're having this issue also like in ALS or MND, where we call this group as pre-symptomatic. But yes, they have this risk gene that follow the behavior of oligogenic high risk gene. However, it's still, we observe like a small number of sample or like people they won't develop the condition. So it's tricky.
So actually, that gets into another whole line of questioning. And actually, I would like to thank the audience. There's like questions coming in the chat and the Q&A, and that's awesome. And that is, so David alluded to high risk when he was talking about, well, you take these high risk people, and ostensibly that comes from genetics, and really, I think Ahmad really sort of identified that solidly. And so, why are large data sets important to you is sort of the leading question I wanted to ask. But I think there's another question, which is, I think we sort of take it for granted now that these large data sets are important, but also how do you manage the issues around reproducibility and harmonization of data when you think about that, when you think about using these to really define risk factors in the clinic?
Well,
David, do you want to take that? You are in a position.
I think we need to be brutally honest here. We're talking about the principles of best practice, not what is happening on the ground. So most medical research is not reproducible, and it's too small, and it doesn't incorporate diverse populations, and so it's not generalizable. And I think we're really underestimating the scale of the challenge and the way in which we need to work and think about these problems needs to be very different. So the more in a recent review that we did in the Demon Network, we identified that more than half of all imaging studies that use machine learning use a single data set, the ADNI study. Now that can't be right. That's like 800 people, which used to be thought of as big for imaging, but you compare that to UK Biobank, for example, and it's tiny. So the resources are slowly improving, but we're not necessarily using what's available now, let alone what should be available or will be available. And it's holding us back. We're not 10 years behind cancer research, in my opinion. I think we're many decades behind. You look at the personalized treatments that they have, they're on another level. And we just have to be more ambitious. So I think we need huge global data to do this properly. And we're just starting. That will require all sorts of things technically and conceptually. And we're not fully grasping that yet, in my opinion. I'm including myself in that. I'm not trying to be pious here. I'm saying this is hard, and this is what we should be talking about. So this is why it's so fascinating.
All right. So actually, I was gonna wait to talk about sort of advances in cloud computing, but I think this has really come up. Somebody just wrote in and said, I do research on hearing loss, and I have huge data sets and other information. What strategies do we have for dealing with that huge data, right? I mean, because we can't swallow the ocean, right? Like we can't be like, tomorrow we're going to be at this technological advancement that David and all of us, frankly, want. And so maybe, and this person asks a fairly specific question. He says, for instance, I use R. Is there a way to use R to analyze millions of data? And yes, or millions of rows of data. And the short answer is yes. There's a lot of ways to answer that question. But Ondrej, maybe we were going to talk a little bit about cloud computing later, but maybe I could ask you to just speak for a few minutes on how you would think about using R for millions of rows of data. Maybe
Sure,
in the cloud, because I think that's a baby step towards the reality that David's talking about.
Yeah, and it's not only about R, as you mentioned. From what I know, and also from my experience from the industry as well as from academia, we know that many people nowadays migrate from HPC to cloud, and this reflects all the need for data analysis. and for actually large-scale data analysis and running their data science pipelines in scale. So in cloud, what I see and think is very, very important and great feature is that like all the like software infrastructure and data, everything is one place. And that is what I like on when working on the cloud. And also the cloud-based technologies nowadays like support many configurations, and that's what you mentioned, Ben. So in some cases, there is no like pre-installed software on some in-house solutions. So in cloud, there is possibility to have something pre-installed and also well maintained. And super example here, I guess, might be Spark technology, also for these hearing analysis and so on. So Spark technology means that this is compatible, or this is meant for distributed data analysis. And this technology and features are actually available on the DNAnexus platform, but it can be also installed by the users on their machines. Definitely. And also just to R, before we jump to R, I would like to also mention, because this is really important aspect of these computations, and these are computational resources like memory, also CPUs, or maybe GPUs, because when we consider working with really large data, we would need to select for us ideal or even the best configuration. So all of these various instance types, and then of course, if we set up, or in case everything is set up properly, so then we can install our softwares and frameworks and packages, and these might be, of course, based, or we can write our programs in R as well as in Python. So again, for example, for Spark, Spark in general supports Python as well as R. So there are technologies how to run the code. So, yes, definitely. So yeah, Ben.
So I think what I want to do, I think that gives an amazing preamble, and if we have time at the end, I really want to come back to concrete steps for doing that. And actually, perhaps if you and Brenton can put some of the webinars we've done on sort of moving to cloud and stuff in the chat, so people can check out some of the more tangible resources there. I mean, I think that you're right. I mean, DNAnexus is one of many places that's working sort of on concrete steps towards these problems. That said, I want to switch gears a little bit, although in the same kind of conceptual framework, and think about conceptual leaps, right? And I think, in my personal opinion, sort of hackathons and prototyping are ways to make sort of technical and conceptual jumps into doing more of the the future of this technological, biological integration with large data sets. And so I actually want to start with David on this one, but then move rapidly to Laura and Ahmad, because they really did a lot of implementation. David did an immense amount of planning with the Alan Turing Institute and others in terms of a pretty large-scale hackathon that a bunch of us were involved in. And David, why? I'm just what I'd like to. I'm hoping you could enlighten everybody on is why it was really worth putting in the effort to launch this really large-scale effort, and what you really hope to get out of it.
Yeah, we just wanted to encourage people to think differently about dementia data. In a nutshell, the Daemon Network is all about innovation. It provides an infrastructure for collaboration. And our vision is that we're going to enhance dementia research and brain health by pulling the right people together. And we're just tired of seeing people doing things in silos. And and we want to play nicely. We want to get people together. We set ourselves ambitious targets for things like the proportion of participants who are from low and middle income countries, because we believe that the people who've got the best ideas are going to be from all around the globe. And, you know, we had participants from 6 continents. So if we wanted to, I could work with Laura and Ahmed, and we could do some great science. But, you know, we need to be more outward looking, you know, and there's no track history in the dementia space of this more computer sciencey hackathon kind of mentality. You know, we come from a traditional background which says, you know, everything's got to be hypothesis driven and slow and careful, and it's just not working, because the problems that we're trying to solve aren't there aren't 12 independent variables and 1 dependent variable. It's just a completely different mindset. And so we need to play, and we need to play with data. We need to meet new people. We need to put teams together who've never met before and are from different countries, and work out how to manage practical problems like time zone differences. So up there, you can see pictures of LA and London, and the whole idea was that we would try and open it up globally by not just having one hub, one physical hub in one time zone. Now that's probably a mistake. It's probably a crazy thing to do, because it's like running 2 events. But it was kind of fun, and it kind of worked, and we need to do more like that. So why did we do it? Because we're not afraid to try things, and we're not afraid to make mistakes, and we want to innovate globally.
So I'd actually like to move to Ahmad, and thank you, David, for that, and the people in the audience seem to resonate with them as well, which is awesome. Ahmad, I mean, you work in the clinic, and I know in the hackathon you had high hopes of really pushing forward clinically actionable things in the hackathon, and I'm wondering if you could talk about that a little bit. You know, I mean, and really in terms of working in terms of things that are in the space that we don't generally consider clinical, and bringing that to the clinical space.
First of all, like, it's a lot of work, but it was worth it. And I think I agree with David. Like, it was, it's taking a step back and do something differently than the traditional way, and where we could make, probably hackathons the quickest way to do research so quickly. Depending on the question you could ask that you are asking, tools can be developed and results can be generated within days. I'm not saying that we reach the polished tools, but this we advance the field so quickly. And I think the most important thing is in the hackathon is the long-term and collaboration. I think getting all these researchers to think about this problem, clinical related problems, and try to brainstorm and create tools so quickly is fascinating. I think NeuroHack was a success. We focus on multiple research fields, one of which is drug development. Multiple tools were created, and it's fascinating how quickly researchers could put multiple resources together, publicly open resources, and create open source also tools for everyone to, for example, find drug from a condition that untreatable conditions. So I think hackathon is a good way also for collaboration. And I think personally, like, hackathon are unique because your skills are the most important aspect. it doesn't matter your sex or your ethnicity, which institute you come from, all of a sudden it's flat, your skills comes and you shine. And no matter you're a professor, you're like a student, all of a sudden we are working as one unit to achieve this task. And this is really important, especially in academia.
So Ahmad, thank you for that. And actually, I just wanted to really extend that. So Laura, you got thrown in this hackathon, and really were sort of boots on the ground for 100 people at a time, working with them to integrate the technical stuff and working with millions of rows of data, and with really sort of a lot of clinical and biological expertise. And you were able to do that on the cloud, which was incredible, and allowed people in LA to get a little bit of sleep, which was also incredible. And so I'm wondering if you could talk about that, like really the experience of being able to jump on to like a cloud platform. This one happened to be a DNAnexus, and being able to work with 100 people at a time.
Yeah, well, I mean, I think I'm going to reiterate what Ahmed's just said about how key it was to having the participants. So they were helping each other. There were some great groups. There were students, there were postdocs. They were all in groups together. They've met for the first time, and they got on and they worked together, and they had access to these different data sets via the platform. So they were working, they were helping each other with code, they were sharing code, they were running each other's code, and they were all able to do that within the Nexus environment. So it was set up so they had their data sets in there, they could run stuff independently, and they were just getting on and doing that, and it's to their credit, I think. So we sort of facilitated it and gave them ideas, but they were there running this, and they were able to do that and go and work on all these challenges and do that within the environment. So
Laura, maybe we can stay with you for a minute. So I was going to ask a question about cloud, and this graphic is, of course, funny and cute, but actually I'm going to skip over this because we've talked a lot about cloud, and really move to thinking about like these international collaborations. And like David touched on this some, but you really, I think, really sort of went directly at it when you were talking about these sort of groups of folks from all over the world, and really throwing in ideas. And Ahmad talked about this some as well. But why is that important? I mean, why is that so important in terms of advancing research and getting us to the state that David was sort of talking about at the beginning?
I think it's key because you get people with different expertise, and it brings different ideas. So somebody will come from one field with an idea that hasn't been used in another field, and you get them chatting and they can apply it there. And we saw that in the hackathon, but you see that within collaboration. But not only ideas and methods, you get a lot of collaboration across data sets as well. So if somebody has a cohort here in one country and somebody has a cohort in another country, you can reproduce and replicate your results. And this is key in reproducibility. So there's a lot you can do with collaboration. So data sets, methods, lots of really important things that come out of collaboration. So yeah, sorry, I've done a very quick overview of what we can do with collaboration. I'll let my collaborators jump in and tell me all the other things I've missed.
David, Ahmad, do you want to expound on that a little bit more? I mean,
well, I think personally, like Laura mentioned, that many resources are now available, like UK Biobank is. I would say like it's one of the greatest biological research resource now and available for researchers. And it's different than what we did before. Like David mentioned that we had smaller data sets, really messy, and also which led to like wrong conclusions, or sometimes research that you couldn't also reproduce. Usually data scientist comes towards the end where the data is very messy and we apply these sophisticated tools, but the data was limited. Now we have this fantastic resource where we can apply sophisticated or cutting edge methods like machine learning, and in using this massive data set and ask and try to answer these important questions. I think it's opportunity also like to get the trust in something like AI and machine learning, for example. Like people developed, it became associated with bad reputation, but now with the massive data sets, we could apply this method and answer it. There are different resources. For example, like I work in Project Mine. It's probably the biggest single disease genetic cohort in the world, and it's focused in ALS and or MND. All these data sets are coming. UK data bank, 23andMe now is a private company with over 8 million genotype available. Iceland Decode, half of population been published, been sequenced. Qatar genome, the whole population is sequenced. So I think we are learning. Methods are getting better.
So,
and you're gone.
Ahmad, directly on that note, there's a question from the audience, and I think it not just for Ahmad, but for the whole panel, which is, so what would each of you like to see from the UK Biobank and other large research studies like that in the next 5 to 10 years to specifically help dementia research? And I'd actually like to break that question into two parts, if we could. And I'd first like to talk about the data and think about what we can do with the data. And then second, talk about technical resources around the data. And I'd like to bring Ondrej into that discussion because I know he has stuff that he really wants to show the audience here. And so, because it's cool, frankly. So why don't we start with the data and then we'll move to really sort of concrete technical steps forward into this sort of new universe of research that David has sort of put in all of our brains. So Ahmad, what would you like to see from the UK Biobank and other large studies?
For me, like for example, like specifically for the disease that or the condition that I'm focused on, the idea that we found pre-symptomatic patient. So that's really unique. We always see this patient afterwards. Now we can see data been collected before and it's longitudinal. It's fantastic way of understanding of events happening before and after. It always thought that MND or ALS have no prodromal stage, but we could answer this type of questions.
So to be direct about that, is that pre-symptomology? Are you reading that from genomic data? Are you reading that from imaging data? Or are you reading that from both?
Both. The unique thing about the UK Biobank now, the microarray data is available. Also, whole genome sequence is available, and also imaging for selected patients. I would say like this is brilliant resource for researchers. And to answer questions, back to David before when he mentioned we have to do clinical trial. This is like almost like a way of randomizing and like doing a snapshot before and after. And for that, like before and developing the disease. For me, this is a fascinating way of doing research. Probably Laura and David and Ondrej will look at this data set differently. And this is why it's also the UK the Biobank is a fantastic resource.
Awesome. Yeah, no, I mean, I totally agree, but like, I'd like to hear from David and Laura about where we can go with this in a data sense. I mean, where would you like to see large data sets go? UK Biobank was mentioned, but also, you know, people are taking transcriptomic data from other sets. I just saw a paper this morning and using those transcriptional QTLs to find new things in UK Biobank and vice versa. And so I'm curious what you guys think, where large data sets need to go.
Well, I think a deeper characterization of the participants is, of course, essential. You're talking about different ways of profiling individuals, sort of from a biomedical perspective. There's also a good amount of clinical data and environmental data, and even things like the geolocation of these people. It's kind of interesting. We're actually just scratching the surface of what we might do with data in combination, I believe. It's amazing. I mean, we've never been able to look at the interaction between a polygenic risk score, so a summary risk score for dementia, and then look at whether a lifestyle risk score interacts with your genetics. I mean, we've always known there might be these kind of interactions, but we've never had the kind of data that would allow us to ask the questions. So this is like the next level to me in terms of where we are. And this just, I mean, there's an explosion of interest in genetic interactions, just as one narrow advantage. But if you don't have the numbers, you can't do it. It's that simple. And so the more deeply characterized they are and the bigger the studies become, the better. I mean, there's literally no downside to having more data.
So David, have you done the PRS experiment yet on UKB?
No, absolutely. We published that in Journal of the American Medical Association. That was our first paper with Biobank in 2019. So that was people like, led by people like Ella Kuzma and Ileana Larida, members of my team, but with international collaborators. Just to come back to, I didn't have all the expertise I needed in terms of polygenic risk score construction, and I didn't have all the expertise I needed in terms of UK Biobank. I could carry on. I didn't have any of the expertise that I needed, really, but I knew how to pull a team together that would have that expertise. And all of our papers that are followed on from that are similar in that sense. But this is just genome-wide data. This is just lifestyle factors. There's so much more that you could do, even with existing data. But was it Oliver's question was about where do we go in 5 to 10 years' time? So this is the cutting edge, I think. I think we can do a lot for precision medicine with what we now have. How could you enhance it further? By coincidence, I was talking to Rory Collins on Monday, who's the PI, the principal investigator of UK Biobank, and he asked the same question. He said, I think this is a great resource for dementia research. And I said, I agree. However, you don't have any case finding. So we only know who develops dementia from medical records and death records. Now, there'll be people out there who will never be diagnosed with dementia. They're doing cognitive assessments, but they're not necessarily ones which are easy to harmonize with other global aging studies. There is neuroimaging, but there's no amyloid imaging, a point that John Hardy, the geneticist from UCL, made. And so all of it could be better, but that's not to say it's not amazing. Of course it's amazing. So 5 to 10 years time, we just need. I still don't think we fully grasp how we're going to need to tackle such a complex condition as dementia, because it's not a single disease. We need to take a lifespan perspective. We know that there are probably at least 30 risk factors for dementia that we know of, the kind of known knowns, if you like. But the current guidelines for dementia prevention emphasize around 10 or 12 for the Lancet Commission, for the WHO. So what are the gene-gene interactions? No clue. I had no clue whatsoever. What are the gene-environment interactions? We're just starting to scratch the surface with this stuff. But is it right?
Can we have a whole nother one of these things just on Mendelian randomization in dementia? I mean,
yeah, absolutely. And in the Demon Network, we are going to have a workshop on Mendelian randomization for these kinds of dementia studies, but Mendelian randomization relies on these polygenic risk scores. The amount of explained variance in dementia is much lower than it could be. So machine learning methods can be used to redo the original GWAS studies, I believe, in studies like UK Biobank. That will give us stronger instruments. Then we can ask different causal questions. But but this is a lot of work. I mean,
So,
but we should be doing it. It sounds fun to me.
So it's interesting. I mean, so when I set out to do this roundtable, I thought that we weren't going to do any technical demos, but actually, I'm going to turn the floor over to Ondrej and ask him to show a couple of very short and simple technical demos about how we're stepping into this world. And Laura alluded to this too, because one thing I want everybody to take home is that this is not an impossible swallow the ocean task that David has put in front of us. I think it's actually a very stepwise task that a lot of people are working on. And so, and we had this fantastic question earlier about analyzing millions of data sets with R. And so I just want Ondrej to show you on the UK Biobank Research Analysis platform how we're thinking about doing this large-scale data. And I'd invite other people that are doing large-scale data analysis on different platforms to think about how we harmonize and how we collaborate, and call us, really. We'd love to work with you. So one thing I'm going to do right now is just turn over the floor to Ondrej for a super short technical demo, and then we'll bring, and I'm going to say, Ondrej, can we limit it to 3 to 4 minutes, and then bring everybody back for really sort of a final Q&A, if that works for everybody. Hopefully, yeah, because honestly, I wanted to write down a bunch of quotes from David, but I also don't want him to scare people. So let's. Let me turn the floor over to Ondrej for just a couple of minutes, so he can show you some of the concrete steps that we've been working on.
Well, great. Can you see my screen?
Yep.
Perfect. So what we have here, first example, is a really example of how we can access and process really large data. So in this example, we are working on the DNAnexus platform. It's not actually UKB wrap, but we are now accessing a product which is called Cohort Browser. So the functionality is really the same for DNAnexus and for UKB wrap. So, and what we have here a really powerful tool, powerful method, how we can create cohorts and how we can compare them. So in this example, we are using synthetic dataset. Actually, we are accessing 100K synthetic data generated from UKB or derived from UKB dataset. And I created here two groups, two cohorts, one group, female group, and then male group. These two groups have approximately, it's approximately balanced group. These are balanced groups. And what we can see here in this data set, we can, in seconds, prepare filtered data. We can filter based on phenotypes, genotypes, and other criteria. So I applied two filters, a filter for female and a filter for male. And what is perfect here, we can add tiles, which basically means tables, other data previews, and also plots. So for example, histograms, scatter plots, and so on. So what I added here for these two groups, I added a volume of gray matter, which could be great indicator of some neurological as well as Alzheimer or other related disorders in this space, in space of dementia. And what I would like to do here, I would like to do one more thing. I would like to do one more tile. And in this case, let's focus on ICD measures and ICD field. So in this case, let's add ICD to these. And here, what is important, everything is one place. So we see here all the possibilities, how to filter, how to access it, how to access data, and also how to analyze data. So here, what we added one more tile, which is really perfect because now it's showing ICD-10 disease classifications. So we can simply access, let's say, dementia group of this ICD information. So here, for example, we now can see, we can compare between female and male and see the proportions, see how many participants belong to each class. So this is perfect. This is the perfect. This is the first example I would like to show. And then what is also possible when we are working on the cloud, typically we can prepare notebooks, which is more for developers or maybe for more advanced users, which can write their own code and also reuse some code, which can be publicly available. So here, as a second example, I created simple notebook for accessing data and showing and visualizing data. So basically, what dementia research or researchers in field of dementia can do is they can run some cutting-edge neuroimaging pipelines and analysis. So for this, or in this example, I downloaded the following pipeline from GitHub repository. It's called AssemblyNet, and I reused some images from this repository. So this is publicly available. So I downloaded this data and I installed in this Jupyter Lab environment library, which is called NightBabel, and showed the data. Data is available in Nifty format for those who are interested in. This is Nifty format, and I was able to describe the data set, describe the data, and plot the data. Like I'm showing metadata as well as individual images and slices of images, volume images, and like individual slices in this case. So yes, so these are the two very brief examples.
Well, thank you very much, Ondrej. I really appreciate you you showing folks all of this. And so I think that's a way to really sort of bring us towards the end and really think about other folks' questions. And I guess, so here's a time for questions. So if you have questions, please type them rapidly into the chat or the Q&A, because we're almost to the top of the hour. But I'd like to really sort of go around the horn here and say, and by the way, any UK Biobank user can use this stuff. Any UK Biobank researcher can use all the stuff that Ondrej just showed you. So check that out, the UKB Research Analysis Platform. Just Google it. You'll find it. And there's a community kind of like BioStars for that too, community.DNAnexus.com. That said, so this is a step forward in terms of the technical stuff we need to be looking at in terms of complex disease. But if we could go around the horn and if we could get a sort of wish list item from everybody in terms of what else do we need to go to the next step? I mean, like maybe I could ask David like a specific question. What do we need to go towards larger scale Mendelian randomization in terms of environmental risk factors? So, and then, yeah, I'd love to hear from everybody.
Well, the lazy answer would be to say more funding. So we can spend £10 billion on a space telescope. So UK Biobank's more in the kind of £200 million kind of range. So what we could do is constrained by that firstly, but given the resources we already have, we can innovate a lot with that, and we can move things forward very quickly, but we'll need to be smart and collaborative about it. So we just need to do that. We need to work differently, and I think there's a lot of appetite to do that. So I'm very positive about this. I'm sorry if I sounded like it's doom and gloom because it's so complicated, but that's what makes it so cool. That's what makes it so interesting and challenging. So definitely get involved. That's what we need. We need the people on this call to get involved. That's it.
So, Laura, can I ask you on a really, I mean, that was an amazingly high-level discussion. Laura, can I ask you from a sort of more boots on the ground perspective? I mean, what do we need technically to get to the next level with genomics and imaging and phenotype integration?
So I was thinking, as David was doing the global one, I was thinking about what I really want next for my data analysis. So I'm really excited about what's coming next within UK Biobank. So there are variables that they're expanding. So they're going to be doing, hopefully we'll get the whole genome sequencing is coming and maybe getting bigger. We're getting, hopefully in the coming years, the proteomics data. That's really exciting. I'd like to see the rest of the primary care records, because obviously we have a third, and we can get a lot of the dementia cases out of there, but we can get the rest of the dementia cases when we get access to the rest of the primary care. So these are really kind of short-term things, but these will really impact on my research in the next sort of 5 years or so, because if I can get that data, I can do more of the research with the dementia ideas. So that's kind of a sort of I don't know whether that's quite what you're after, but that's really what I want. So that's what I'm really hoping for, and I'm really excited that we'll be doing next within UK Biobank. So those new sets, expanding those sets and getting more omics is really what I'm interested in and what I'm excited to get working on next.
I mean, frankly, that motivates me to work in the evenings, because I'm trying to get notebooks out for people to do proteomics analysis when the tranches of data come out July 1st. So absolutely, because I mean, there's a lot we can do in terms of pharma validation there, I think. And last word, I guess, goes to Ahmad. Ahmad, where does this go clinically? Like, I mean, how fast can we start getting things into the clinic if we do all these things that Laura and to some extent David are talking about?
Yeah, I agree with what David is saying about collaboration. Open science is the only way forward. Also, multiple level of data. As we see, like genomics is not enough. It's fantastic. Like we need to build a more risk scores or like scores based on multiple omics data. I think this is the way forward. It would be great to have this available for in Biobank. However, like I think this amount of data are now available for clinicians also. So like we are not prepared for this. Probably we need more of a clinician that can understand the implication of this large-scale data set, and how, for example, now we have patients coming also with their genome being sequenced, or also like data available, but we don't have enough clinicians to understand this data set or how to work with it. Probably now we have to think about tools that can make this simple. And to take this data set, do the analysis, and provide summary of this the analysis in a way that clinician can understand, and also so also can impact patients' life. Hopefully, we can reach that.
All right. Yeah, I totally agree. And yeah, I would just add to that I think, for things like GWAS, clinicians understand covariates sort of intuitively, whereas as data scientists, we're always
you are giving your doctor a lot of credit when you think that your clinician doesn't know what GWAS is. So like you have to understand, like we need to train next generation clinicians of that, for example, I would suggest data science part of med school, and also having clinicians that specialize in data science, that's like this, fellows, rather than just you have to spend 10 years doing neurology, and after that you do like 4 years to 6 years in PhD. Then we need to think differently. How can accelerate this?
Well, I want to say yes, and I want to say that's something we should take back to the UK Biobank, or collaborators at the UK Biobank, perhaps, that we want to start teaching some workshops in some medical schools. That literally may have legs. I mean, we can see. So with that, I'd like to thank everybody. And I'd really like to thank all the attendees for hanging on. I mean, we didn't really get people dropping off until a couple minutes before the hour. And now we're past the head of the hour and most attendees are still on. So thanks for hanging on. Thanks for your interest in this. And really, again, reach out for collaborations if these are things you're interested in. And if you want technical help doing stuff, reach out. There's a lot of resources here, and we love talking to people and collaborating with people. So thanks again, and we'll hopefully have another one of these soon. All right. Thanks, everybody.